Right, let's dive straight into this explainer and open up the forensic case files. We're going to see exactly how we solve mysteries that the scalpel simply cannot. Welcome. Speaking as a forensic pathologist, my entire job has historically been rooted in what I can see, touch, and measure on the autopsy table. But medicine is evolving, and today we're looking at the absolute frontier of post-mortem investigation, the molecular autopsy.
You know, as a forensic pathologist, there's literally nothing more haunting than an unexplained death. Imagine this. You have a previously healthy young person, a sudden tragic event, and when we start looking for a cause, maybe a structural defect, a massive bleed, or a blocked artery, we find absolutely nothing. The heart is perfectly normal. The brain is completely intact. This terrifying blank space is exactly what brings us to the cutting-edge science of the molecular autopsy, where we literally have to shift our gaze from the macro down to the microscopic.
So to make sense of this, we'll structure our investigation today across 6 critical phases. Beyond the traditional autopsy, the MDMA metabolism mystery, decoding DNA's technical workflow, pharmacogenomics, the ethics of genetic risk, and finally, closure for the living.
Let's move right into Section 1, Beyond the Traditional Autopsy.
Now, in a traditional autopsy, we can easily spot visible structural defects. We're talking things like an epicardial hemorrhage, a bleed on the heart, or maybe a ruptured aorta. But the molecular autopsy, it searches for what we cannot see. It's looking for things like genetic channelopathies, which are essentially invisible flaws in the heart's electrical wiring that cause fatal arrhythmias, or tiny variations in CUP450 liver enzymes that dictate exactly how our bodies process chemicals.
And here is the truly, absolutely crucial point. Nearly 30% of sudden cardiac deaths in young people remain completely unexplained after a traditional autopsy. 30%! That's almost 1 in 3 cases where the heart appears structurally normal. So without a physical explanation, we have to rely on a panel of genetic markers for conditions like long QT syndrome and other primary arrhythmogenic disorders. Because if we don't look at the DNA, a third of these grieving families are left completely in the dark.
That brings us to Section 2, the MDMA metabolism mystery.
Let's analyze a real case at the cellular level. We have a 16-year-old girl who is insulin-dependent and tragically died after taking MDMA. Her histology reveals severe brain swelling or edema following a hyponatremia crisis, basically a severe drop in blood sodium. What happens is MDMA can trigger a massive release of antidiuretic hormone and induce extreme uncontrollable thirst, a condition we call polydipsia. She drank so much water it fatally diluted her blood sodium levels.
But then, in stark contrast, we have a second victim, another 16-year-old girl who collapsed at a rave and died hours later. But her body reacted completely differently. She experienced fatal hepatic failure and epicardial hemorrhage. And here's the real kicker. The extent of liver injury or brain swelling in these cases doesn't always correlate perfectly with the dose of the drug ingested or even the blood concentration. We have 2 young women, the exact same substance, but entirely different physiological cascades, leading to death.
So why does this happen? Well, it all comes down to individual invisible vulnerabilities. Perhaps it's a deficiency in the CYP2D6 liver enzyme, which is basically the body's chemical processing plant that breaks down the drug. Or it could be an unpredictable impairment of serotoninergic neurons, immense oxidative stress, or that extreme fluid intake we just talked about. Ultimately, your genes are setting the stage for exactly how your body is going to react to a chemical insult.
Let's transition to Section 3 and see how we actually find these genetic anomalies. Decoding DNA's technical workflow.
Finding a gene mutation in a sudden death case is incredibly challenging. But here is step-by-step how we pivot from traditional organ inspection to modern molecular screening. First, we secure a post-mortem blood or tissue sample. Next, we carefully extract the DNA. Then, we subject that DNA to some highly advanced sequencing techniques. And finally, and this is for sure the most difficult step, interpreting those genetic sequences to definitively link a specific mutation to the cause of death.
The tool we use for this is called whole exome sequencing, and it acts as our ultimate diagnostic net. Instead of just guessing and checking one gene at a time, which would take forever, this technique screens the coding regions of all 22,000 genes at once. It's an absolute game changer. It allows forensic pathologists to detect variants related to almost all major genetic diseases from just one single sample, catching those microscopic mutations responsible for sudden fatal arrhythmias.
Okay, moving on to Section 4, pharmacogenomics and the fentanyl case.
Pharmacogenomics is where genetics marries pharmacology. By understanding a person's exact genetic makeup, we can finally explain that vast, baffling variability we see on the autopsy table when it comes to drug toxicity. It literally explains why a seemingly safe, therapeutic dose for one patient acts as a lethal poison to another.
It turns out specific variations in our genes critically alter drug metabolism. Let's take fentanyl, for example. If an individual inherits 2 copies of a specific mutation, a homozygous mutation, in their CYP3A5 gene, their ability to process fentanyl is severely impaired. The same goes for methadone. Tiny genetic typos, known as single nucleotide polymorphisms, can turn someone into a poor metabolizer. So genotyping tells us if a routine dose essentially became a fatal overdose simply because that person's body was genetically incapable of clearing the drug.
Now let's tackle Section 5, the ethics of genetic risk.
This is where science hits the real world, and we have to balance the cold exactness of data with true compassion. Families often struggle with the terrifying decision of whether to uncover their own inherited risks. Currently, there's often no legal requirement for a medical examiner to get family consent before running a molecular autopsy. So families might suddenly be burdened with the terrifying knowledge that they possess a fatal genetic mutation, all while their loved one's DNA is retained infinitely in a lab. Yet, as pathologists, we feel a profound medical duty to warn surviving relatives if a fatal, inheritable risk is discovered.
To put the sheer weight of this data into perspective, discovering a fatal gene in the deceased means half of their children likely carry it too. Over 95% of these sudden arrhythmic death syndrome mutations are chromosome-dominant traits, so if we find it in the deceased, their siblings, their children, their parents, they are all instantly thrust into a high-risk category. At this point, the autopsy is no longer just about the dead. It becomes intimately, critically tied to the living.
Which brings us to our final section, closure for the living.
While our forensic work obviously always starts with the deceased, our most profound duty is actually to protect and provide closure for the people they left behind. You can think of a molecular autopsy as an early warning system. By identifying the exact genetic flaw that caused a sudden cardiac death or a fatal drug reaction, surviving relatives can be screened, monitored, and given preventative treatments like implantable defibrillators that will literally save their lives. It replaces agonizing, terrifying uncertainty with actionable clarity.
I want to leave you with a final, provocative thought regarding the heavy ethical weight of uncovering this invisible evidence. If you were in their shoes, navigating the sudden, tragic loss of someone you love, would you want the pathologist to look into their DNA, knowing that the results might just rewrite your own medical future?